So we cannot talk about immunity without mentioning the major histocompatibility complexes. There are two actually, so we're going to review those briefly. So basically when we encounter a pathogen, there are two ways that we can deal with. For now, let's say there are two ways. Okay, number one is when the pathogen is phagocytosed. Okay, in this case, the pathogen will reside in a phagosome that will merge with the lysosome and will be digested. Okay. The other way is if the pathogen resides into the cytoplasm. Okay, so these are some intracellular bacteria like Listeria monocytogenes, for example, or viruses. Okay, so viruses will inject their content, whether it's DNA or RNA, inside the cytoplasm and use cellular machineries to reproduce. Okay, basically. So let's review this, what's happening. Okay, so we have a virus here that made it from the outside, made it inside of the cytoplasm, and it will, the viral peptides and proteins will be digested by the proteasome. So what is a proteasome? It's a whole enzyme, okay? It's a cluster of stuff. It's basically, think of it as a housekeeping genes, okay? It's disposing stuff that are not needed. So the obtained peptides, okay, then via TAP, okay, channel are going to go into the endoplasmic reticulum and will be associated with MHC Class I molecule. So MHC class molecule, it has a couple of subunits, alpha 1, 2, and 3, and beta 2 macroglobulin. Okay, so we are assembling this complicated structure that eventually is going to go to the Golgi apparatus. It's going to be wrapped into the secretory vacuole, and it's going to be put outside of the cell. And when that complex is expressed, okay, it will recruit, basically, it will be recognized by cytotoxic CD8 positive T cell. And CD8 positive T cells will activate a granzyme and perforin. It's punch a hole here and it's going to kill that cell. So basically, MHC class I is a way the cell to wave a red flag and say, I'm infected, kill me, basically. Okay, so the problem is that some viruses, for example, cytomegalovirus, has a ways to actually downregulate MHC type 1, which means it will be missing on the surface here. And if it's missing, it will not be able to recruit CD8 positive T cells. So what's going to happen, we develop an additional mechanism, okay, in which we activate NK cell, natural killer cells. And natural killer cells are going to be sniffing out cells which do not have MHC class I. If they don't have it, there is a high chance, okay, that it's being downregulated by the virus. So NK cells are still gonna kill the infected cell.